Okay, so the app closed before, so I'll just repeat what I said. Cellular adaptations are very important, and the most important thing to remember here is that all of these are reversible in nature. That means the difference between dysplasia and cancer is that dysplasia can be reversible, but then cancer is always irreversible. Now, there's a couple of ways you can remember what all of these words mean. The simplest way is that you need to remember that trophy means size, and then plasia means number. Okay, so trophy means size. So if you have hypertrophy, that means increase in size, and then hyperplasia. So number is plasia, and then hyper is more. So more number, increase in number. Now atrophy, A means no or less, and then trophy means size. So it means a reduction in size. Then metaplasia, meta means change, and then plasia means again number. So change in the number, which kind of essentially means that there is a change in cell type. Finally, dysplasia. Now, dys means something dysfunctional, and then plasia means again number. So it's an abnormal group of cells. So that's how you can remember the name, and it's pretty easy, right? So based on the name, you can figure out what it means. But then the question comes, why is this page of pathology so important? It's very important not because of the actual, yeah. So it's very important not because of the context, sorry, not because of the material that's present in the page, but it's more about the context. And like, if you understand these principles, then you can understand why certain things happen and what they can lead to. So let's keep this more interactive, and I'll ask you questions about this. So let's say you want to increase the size of a cell. Okay. So if you want to increase the size of a cell, what would you need? How can you increase the size of a cell?
Proteins,
Yeah, so you'll need proteins, basically
water retention.
water. Okay, yeah. So the idea is if a single cell is very small and you want to increase its size, firstly you'll increase the cell membrane so that you can put in more stuff to increase the size. Okay, so you increase cell membrane that is made up of proteins. That's one. After that is done, you want to increase the amount of organelles. Now, one mitochondria won't be enough for a big cell, so maybe you need two or three. So now you're making more mitochondria, so you're increasing the number of organelles. Let's say water is one more because the inside of the cell is cytoplasm, which is majorly made up of water. So you want to put in more water. So all of these things here, they only give you proteins and organelles, and the reason why they give you these two things is because proteins, they make up the cell membrane and the associated proteins within the cell, and then the organelles, because to sorry, to use the entire cell, to have sufficient function and to keep the cell alive, you need more organs because now it's a bigger cell. Now, why does hypertrophy happen? Okay, we'll take an example of, let's say, a simple muscle cell. When you go to the gym, let's say a normal muscle is 10 centimeters in size. Now, when you lift a lot of weight, your muscle says that I need to kind of grow bigger so that I can lift this weight more easily. So, any kind of stress which is placed on a group of cells will cause them to increase in size. On the other hand, if you remove the stress, then the size will go down. Now, based on this, let's take the example that they've given us. They've given us cardiac hypertrophy. So now, what could be a reason why you have cardiac hypertrophy?
Stress.
Yeah.
For
So
example, hypertension.
yeah. So anything which increases the stress of the cardiac cells.
Also
Now,
increase with age.
yeah, with age, but even with age, right? The reason why you have cardiac hypertrophy or like yeah, cardiac hypertrophy with aging is because your arteries they get a little stiff. Now, if your arteries
Okay.
are more stiff, then your heart needs to pump harder. And if your heart needs to pump harder, means it's doing more work. And if your heart is doing more work, then you need to increase the size of the cells. So that's why any kind of stress that the heart is
Okay.
facing will cause hypertrophy. Now, this is very interesting. Heart can only go heart can only undergo hypertrophy, not hyperplasia. Why do you think so?
Mm,
Why do heart cells not increase in number?
I think cardiac cells are terminals, right? They don't go under they don't undergo mitosis.
Yes, because they're permanent tissue. So heart
Oh,
doesn't go like undergo mitosis. Which other ones do you know?
brain.
neurons.
Brain's another one.
Neurons, yes. Any other ones?
Striated muscles.
Striated muscles, they can. Yeah, okay. So they can do regeneration, not replication. Yeah, okay. Striated muscles. What about the liver?
Skeletal.
Liver regenerates.
Yes,
Yeah,
liver regenerates. Liver regenerates a lot. You can have like 50% of the liver and it will regenerate the other 50%.
right.
And then striated and skeletal muscles, they're the same. Yeah.
Okay.
So all of these cell types, so cardiac, nervous, striated muscles, they would undergo hypertrophy, not hyperplasia. Why? Because they're permanent tissue. But now, what about, let's say, the endometrium? The endometrium, because it's constantly growing, so you'll see both the things happening together. You'll see hypertrophy and hyperplasia together. Now, to increase the number of cells, what's the most important process that needs to happen?
Okay.
So, what's the most important thing if you want to increase the number of your cells?
Stem cells.
Stem cells, and the other one is replication or mitosis. So, hyperplasia has increased mitosis. Here, it also says increased production of stem cells, which will ultimately lead to increased mitosis. So, basically, you want to increase the number of cells. Now, let's say there is okay, this one here is actually interesting about endometrial hyperplasia. So the endometrium, it's dependent on estrogen. Okay, so whenever you have estrogen, the endometrium grows in size. Now, what if I give the endometrium too much estrogen? It will increase in size. But now
Yes,
what?
it will.
Yes, but then if it increases in size too much, then do you think there is a problem if we give the endometrium too much estrogen?
Yes,
Yes.
it would hinder to its normal functions.
Yeah. But do you think it increases the risk for something?
Uh, Cancer.
Yes.
Endometrial cancer.
Yes. Endometrial cancer is because of an increase in estrogen. So it's not only the metaplasia which gives cancer, but even hyperplasia can give you cancer. So any kind of cellular adaptation, if it's too severe, will lead to cancer. Now, that was for hypertrophy and hyperplasia. Now, what about atrophy? You're decreasing the actual mass. Here, they've defined it as mass of the tissue. And if you want to decrease the mass of the tissue, then you will have to do both the things. You'll have to decrease the size and also you'll have to decrease the number. Now, if you want to decrease the size of a tissue, or if you want to decrease the size of a cell, so then what would you do? You want to remove the excess protein, right? So you want to shrink the cell membrane. Maybe you want to get rid of some organelles. So which mechanism in the cell can lead to destruction of protein?
Cytoskeleton.
So cytoskeleton is the membrane. So we do want to remove that membrane to make it a little smaller. But the actual mechanism is actually this one. So the ubiquitous proteasome pathway. Now, how does this work? There's a small molecule called ubiquitin. This ubiquitin goes and attaches to the protein that you don't want. Now, when ubiquitin attaches to this protein, there is the proteasome which comes along and then dissolves the protein. So that is how it works in like a short summary. Ubiquitin marks the protein and then proteasome degrades the protein. That is how you reduce the amount of proteins. And if you can reduce the amount of proteins, you're basically reducing the size of the cell. Now, how do you reduce the number of cells? There's two ways. So what do you think? What are two ways in which number of cells are reduced? Hello.
Yes.
Yeah.
Mm,
Oh, okay. So Mahin wrote apoptosis is one, yes. But what's the other way? Okay, maybe not normal, maybe pathologic. But what's the other way in which you can reduce the number of cells? Necrosis, yes. So
gang.
apoptosis and necrosis, both of them can reduce the number of cells. Now, if you don't want inflammation, if you don't want pathology, then apoptosis. It's natural and it's programmed. Yes, Devanshu.
Yeah, in gangrene, is it necrosis, liquefaction of
Yeah,
cells, and or is it right? Is it necrosis?
yeah. Gangrene is necrosis.
Okay.
So it's gangrenous necrosis.
Okay. Thank you.
No problem. So now there are two ways. You have apoptosis and necrosis. Apoptosis is the programmed cell death. That means your body actively tries to avoid inflammation, and it's done within the body, through the body, for the body. So it's very democratic. On the other hand, necrosis happens because of some kind of injury. So this could be because of oxidative damage. This could be because of trauma. It could be because of decreased blood flow. Any of these. Anything pathologic will give you necrosis. Anything physiologic will give you apoptosis. Now let me give you an example. Blood vessels, when they're initially made, they're one solid tube. To convert them into a hollow lumen like pipe, the center portion of the blood vessel, it undergoes apoptosis. The entire center portion undergoes apoptosis. Now if this were to be necrosis, you can't control necrosis. Maybe there is some part in the blood vessel which just also dies. The surface of the blood vessel dies because of necrosis. So now you'll have bleeding. So your body wants apoptosis because it's very controlled and very specific and it's physiologic. Necrosis, very bad. And there are multiple types of necrosis that we'll study later on. Next is metaplasia. Now, metaplasia, as we said, is changing of cell type. Now, this change happens because the stress that is put on the cell is not what it's usually handling. Okay, let me give you an example. Let's say, okay, I'll give you the normal example. Let's say someone starts smoking. So, normally, your mucosa in the trachea, its only responsible or its only job is to allow air to pass through. So that's why it's producing some mucus, it's got the cilia, all of these things. But now, because smoke is being exposed to the epithelium, that epithelium changes into squamous epithelium. Now, why squamous? Because squamous cells are much better resistant to smoke. So that's why your body changed from normal epithelium, which is um, columnar with goblet cells and ciliated. From there, it changed to squamous epithelium. Why? Because you had to change the type of stress that is placed on the epithelium. Now, there are two very important metaplasias that you need to remember. The first one is Barrett's esophagus, and then the second one is tracheal. So, this part here, let me see if they've added Barrett's esophagus. Oh no, they haven't added the exact translocation for esophagus. Okay, no problem. Now, uh, these are the two you need to remember. And usually, metaplasia does lead to cancer. So, not directly. It will be metaplasia, dysplasia, and then cancer. Now, there is one cancer, one more metaplasia that you need to remember is that in the transformation zone of the cervix, you have squamous metaplasia followed by squamous cell carcinoma. So, what do you think the reason for this squamous cell carcinoma would be?
Reason or cause?
Reason, cause,
Are you
either one.
talking, asking about the cause? Okay, yeah, it's, I think HPV.
Yeah, HPV is correct. So, squamous metaplasia and squamous cell carcinoma in the transformation zone of the cervix would be because of an HPV infection. And then which specific HPV subtypes?
16 and 18.
Yes, 16 and 18. Now, those are the three important ones, but there are multiple different metaplasias. Here they talk about myositis ossificans. So even if you were to break the name, it says myo, so muscle, inflammation, and ossify. That means turns into bone. So basically, there was some kind of muscle inflammation which led to bone formation in the muscle. That is, yep, it says bone formation within muscle after trauma. So simply based on the name, you can assume what the metaplasia is. Here, it's myositis ossificans after trauma. Okay. Finally, we have dysplasia. Now, dysplasia is dysfunctional cells, disorganized cells, bad cells basically. These are precancerous. They're not cancerous yet. They can still be reversed, but they're very disorganized. Here they might have, yeah. Oh no, that's neoplasia. Yeah, you see dysplasia. You change the structure of the cell. You change the size of the cell. Maybe the functioning of the cell is changed. All of these things. The entire cell type is changed, or I mean, it's still the same cell type. It's just the structure and the size is changed. And dysplasia has a very high risk for developing into cancer. And then once it develops into cancer, then it's irreversible. And because it's irreversible, you would have to do some kind of treatment. usually chemotherapy, or you can surgically resect these. Now, let's see if they've given us some additional. Okay, so this nuclear change, I would say, is very important here. Yeah. So you do need to remember that there is an increased nuclear to cytoplasmic ratio. That means the nucleus is much bigger compared to the cytoplasm. And when we study about cancer, you'll see that this is also seen in a lot of cancer cells, that the nuclear cytoplasmic ratio is increased. And along with that, you have you have clumped chromatin. And chromatin is basically the non-condensed form of chromosomes. So any questions with these cellular adaptations?
So I think these questions usually integrates these kind of cellular adaptations, right? Like in examples, are they asked individually?
No, they won't ask individually, but they'll integrate it with other things. I would say that you'll never get a specific question saying like, oh, is this process hyperplasia or atrophy, or what is this specific process? They'll usually give you something which includes all the things together.
Okay.
So you'll have to use these concepts to actually get the answer, but they won't ask you about these concepts itself.
Okay.
Yeah,
Okay. Thank you.
no problem. Okay. So any more questions about the cellular changes, adaptations?
No, Doctor.
Okay. Then we'll go to. Oh, okay. Yep. So next we have cellular injury. Now, cellular injury, there's two types. One is reversible, one is irreversible. What's the difference between them? What do you guys think? What's the difference between these injuries?
Their names.
I mean, their names too, yeah. But one of them is irreversible, one of them is reversible. That's the main difference. Now, except for the name
Is
and the difference. Yeah.
ischemia versus infarcting,
Yeah.
like a tissue
Ischemia versus
infarcting.
infarction. Yeah, that's true. But here, there is a couple of things. If you remember that, then the other things you can figure out. I would say irreversible injury. This part here is the most important. That there is cellular swelling. On the other hand, for irreversible injury, you need to know that there is breakdown of the plasma membrane. Now, let's say a cell swells up. It just increases balloons in size. Sorry. Let's say the cell just balloons in size. Okay. So the plasma membrane, it would become flat. Usually the plasma membrane has some invaginations, but now that won't happen, it would become flat. After that, if it keeps on growing, then you'll have these bleb formations. So you'll have like small, I think, outdentations. Yeah, you'll have like small bubbles forming on the membrane, something like this. See, here they have these blebs. So you'll have those bleb formations. Except for that, because there's so much increased pressure, there's so much fluid which is being built up, you'll have the ribosomes which separate from the ER. Additionally, the chromosomes, you'll have like clumping. So many changes. All of them because there is swelling of the cell. Now, yeah, Devanshi.
Plabbing is only in reversible or in both?
Oh no, only in reversible. In
Okay.
irreversible changes, there is something completely different which happens.
Okay.
Yeah. So yeah. So you have these blebs which are formed. Then you'll have the loss of the ribosomes from the ER. And if you have loss of the ribosomes from the ER, then you're reducing your protein synthesis. All of these things happen. But the cell is still alive. The cell still can be turned back to normal if you give the cell energy. Now, this the mechanism of why reversible injury happens is very important. Basically, we want to know that the sodium potassium pump will stop working. Now, if the sodium potassium pump stops working, what do you think? What will happen? What electrolyte will increase in cell, which would be decreased? Oh, hello.
Yes,
Yeah.
Dr. Taha.
So, like in the sodium potassium pump, which electrolyte will go out? Which electrolyte will come in?
Potassium would three and three.
Yeah, 3 2.
Potassium would, yeah, it was 3 2. Potassium would come in and sodium would go out. 2 sodium and 3 potassium.
Yeah, potassium will come in and sodium goes out. Yeah. Now, if you stop the pump, if you stop the sodium potassium pump, then which electrolyte will increase inside the cell?
Potassium, because it is coming inside the cell.
Um, yeah, but sodium will increase in the cell, right? Not decrease,
Sodium.
increase. Yeah, sodium will
Oh,
increase.
because it cannot go out.
Yes, so sodium will increase in the cell. Now, we remember the logic that water follows the solute. So, if there is a lot of sodium inside the cell, where will water go?
Into the
Into
cell
the
become
cell.
Inside.
swollen.
Yes. So, that is why you have the swelling of the cell. Now, that is for a reversible injury. Now, think about a balloon. I know we're shifting a little, but think about a balloon. If you fill it up too much, if you like blow a lot of air in the bubble, in the balloon, what do you think ultimately will happen to the balloon?
You'll
It
burst.
will burst.
Yes. Same thing happens in irreversible injury. Because there is so much water which is going inside the cell, it just bursts open. Now, if the cell is bursting open, everything kind of goes bad. You have mitochondria which gets damaged, you have lysosomes which get damaged. Now, lysosomes, they have these very corrosive enzymes, proteases. So, if the lysosome gets damaged, it will start eating up the cell itself. The nucleus gets damaged. And if nucleus is damaged, then the cell cannot replicate. Cell dies out. Now, see, this membrane broke down. Now, there is one very interesting thing. In the cell, there are a lot of enzymes, the cytosolic enzymes. In the heart, it's troponin. In the liver cells, it's AST and ALT. So, if there is a lot of cell, if there is a lot of enzyme in the cell, and the cell breaks out, where will the enzyme go?
Blood.
Yes, in the blood. Now, if let's say you want to figure out if there is irreversible injury, you will check the blood for these markers. So that means if you ever see troponin in the blood, you can say that one cardiac cell has died. There is irreversible injury, there is breakdown of the membrane of the heart cells. That is why you have troponin outside. There is some kind of damage to that cell, irreversible damage. Now, finally, we have this part. So, nuclear degradation, it happens in three phases. These are pycnosis, karyorrhexis, and karyolysis. And sadly, you'll have to remember these names. But pycnosis is condensation of the nucleus. Karyorrhexis is fragmentation, so it breaks up into smaller parts. And then karyolysis is just the dissolution, so it just completely dies off. So you condense the nucleus, you break it into smaller fragments, and once you've broken it into smaller fragments, you dissolve the nucleus. Yeah. So that is for reversible and irreversible injuries. So any questions with these topics?
No,
No.
doctor.
Okay, perfect. So I think
We'll
we'll.
ask tomorrow after solving questions.
Yeah, I was thinking like there would be a lot of questions, so that's why I said okay, we'll only cover this part. But I think we covered it too quickly.
Yeah, because question arises when we start solving questions.
Yeah. So
It happens with me like that.
yeah, hopefully we'll solve some questions soon. Okay. Okay. So I was thinking because I wanted to do a kind of differential for these. Okay. So I mean we already talked about it. Apoptosis is programmed cell death, and necrosis is damage, some kind of trauma, pathologic cell death. Now, because apoptosis is programmed, you need to remember that apoptosis requires energy. Apoptosis is dependent on ATP. On the other hand, necrosis happens because of a lack of ATP. That's the first thing. Next thing is this part. Apoptotic cells will almost always be deeply eosinophilic in nature. Sometimes, even in the question, they'll just show one random eosinophilic cell in a whole group of normal cells, and they'll ask you what process is happening in this specific cell. And the answer would be that, yeah, there is apoptosis which is happening in the cell. So there is deep eosinophilic cytoplasm. Now, finally, if a cell dies because of apoptosis, it's always phagocytosed. Oh, yeah. The cell is always phagocytosed by macrophages or by neutrophils after it's been dead through apoptosis. Okay. Now we need to figure out how does apoptosis happen. So there are two major ways. You have the mitochondria, and then you have the extrinsic pathway, which is divided into like two ways. Actually, wait, and oh yeah, this one. So extrinsic pathway has two forms. Now intrinsic pathway. For the intrinsic pathway, right? What happens is initially you have some kind of damage to the mitochondria. This damage to the mitochondria, it eventually leads to destabilization of the mitochondrial membrane, which is because of a protein called as BCL2. Now, if the BCL2 goes out, it allows for the sorry, if the BCL2 goes out, then you have increased release of cytochrome C into the cell, and this cytochrome C it activates caspases, and then caspases start the apoptosis process. So these are the three things you need to remember. One, BCL2 gets damaged. Because BCL2 is damaged, you have cytochrome C, which is released from the mitochondria. This cytochrome C activates the caspases, and then the caspases activate the proteasomal enzymes, which then lead to breakdown of all the inner structure of the cell. Now, obviously, I will let you guys read all of these things because tomorrow, if you have any questions, then we'll discuss all of these again because these are very important topics. So I'm just giving you a brief overview today, and then tomorrow maybe you can review them and then we discuss them again. Yeah, Devanshi.
Uh, can you please explain the term pycnosis and karyohexis like it's in the apoptosis first paragraph?
Oh, yeah.
I have forgot
So,
those things.
wait, where did it go? Okay, very well. So, pycnosis,
Last,
karyorexis, and karyolysis. Okay, pycnosis is when you have shrinking of the cell.
yeah.
Okay, so you have shrinking of the cell in pycnosis, not the cell, the nucleus. So nucleus shrinks, pycnosis. Now, karyorexis is when the condensed nucleus gets broken down into
Mhm.
smaller parts. Okay,
Okay.
karyorexis is when condensed nucleus gets broken down into smaller parts. Finally, you have karyolysis. Karyolysis is when the smaller parts degenerate or they dissolved, they get broken down.
Okay.
So once again, pycnosis is condensation, karyorexis is fragmentation, and then karyolysis is resorption or dissolution. Yeah.
Okay.
Yes,
Thank you, Dr.
no
Taha.
problem. Yep. So, and once again, for the inner mitochondrial pathway, you have BCL2, which maintains the mitochondrial membrane. If there is some kind of damage, then the mitochondrial membrane BCL2 protein stops working. So then the mitochondrial membrane leaks out cytochrome C. Cytochrome C activates caspases, and then the caspases they ultimately cause the destruction of the cell by activating proteasomal enzymes. Yep. So any questions with this one? The intrinsic pathway.
No, doctor.
So then next we have the extrinsic pathway. Now the difference between the intrinsic pathway and the extrinsic pathway is that intrinsic something happening within the cell. So BCL2 got messed up because something happened inside the cell. And then extrinsic, sorry, and then extrinsic pathway is when a different cell, so usually a T cell or an NK cell, that cell binds some kind of receptor on the cell surface, and that leads to apoptosis. There are two different receptors here. The first one is the Fas, and you need to remember this that the Fas and Fas ligand that can cause apoptosis, and the second one is the TNF alpha receptor. So both of these, how this works is you have a cell, and the cell has a the cell has a receptor, and the T cell or the natural killer cell has the ligand. When the natural killer cell fixes the ligand on the receptor on the target cell, that cell would internally start apoptosis because of the receptor. Additionally, one thing to remember is that the process is still the same. That yeah. Ultimately you need to activate these caspases. So when you have binding of the Fas cell or the TNF alpha receptor and the ligand, you're still ultimately activating the caspases. And when you activate the caspases, they then eventually lead to breakdown of the cell. Now here they've given us a rather interesting condition, which is autoimmune lymphoproliferative syndrome. Now, what happens in autoimmune lymphoproliferative syndrome is that you have a defective extrinsic pathway, apoptotic extrinsic pathway. Now, if you were to think which immune cell most, and this is actually a side topic, but which immune cell undergoes the most amount of apoptosis?
B cells.
Yeah, so B cells, T cells. For T cells, we have the negative selection. So that is all. So sorry, not the negative. The positive selection, the negative selection. So one you have apoptosis, one you have energy. So both of these things. So I mean, technically, I should say that all the immune cells undergo apoptosis eventually. Not right now, but maybe eventually. They all undergo apoptosis. Yeah. Now, what happens in autoimmune lymphoproliferative syndrome is basically you have these lymphocytes which are not actually functional, and they're very self-reacting. That means they attack the body itself. So you have lymphocytes 1, they're attacking the body, and they're growing so rapidly that they're causing like enlargement of the organs. Now, because these lymphocytes, they're also attacking your own cells. That's why you'll see anemia, you'll see leukopenia. Basically, all the blood cells they'll go down. So, what would be the features? Let's recap. You'll have lymphocytosis, that'll be one, very high lymphocyte levels. You'll have hepatosplenomegaly or lymphadenopathy because they're growing very quickly inside the cell. And third, you'll have cytopenias. So you'll have anemia, leukopenia, maybe thrombocytopenia, all of these things.
Okay.
And then finally, we have the perforin and granzyme pathway. Now, this pathway, which cell do you think uses this pathway or causes death because of this pathway?
Cytotoxic and natural
Yes.
killer.
So both of those cells. Now, if you use the names right, perforin, it perforates the cell membrane. That means it creates a hole. Sorry, it creates a hole inside the cell membrane. And then granzyme is the enzyme which enters the cell and then activates caspases, which then ultimately leads to damage to the internal components of the cell. Perforin perforates and granzymes cause activation of caspases, which then break down the cell. Yeah. So, any questions with apoptosis?
No, doctor.
Okay. So then let's end for today. I will see you guys tomorrow. Tomorrow we'll do necrosis. Maybe we can also